For over 300 years, we were told that human beings could be sorted into neat biological boxes. Races, each one supposedly carrying its own distinct genetic package. Skin color, bone structure, blood type, even intelligence. All of it, we were told, traced back to which box you were born into. Doctors believed it. Governments built entire legal systems around it. Scientists themselves spent generations trying to prove it. Then, finally, researchers did something nobody had ever been able to do before. They opened up the human genome itself and looked, letter by letter, at the actual code that makes us who we are. And what they found didn't just challenge the old idea of race, they quietly, methodically, and completely took it apart. To understand how big this moment was, you have to go back to where the idea of race actually came from. It wasn't handed down by nature. It was invented by people, and relatively recently in the story of our species. In the 18th century, European naturalists began classifying humans the same way they classified plants and animals, sorting bodies into a handful of categories based on skin color, skull shape, and geography. Carl Linnaeus, the same man who gave us the modern system for naming species, split humanity into groups he described using language soaked in the assumptions of his era. Later thinkers built on that foundation, measuring skulls, comparing facial angles, and constructing elaborate hierarchies that conveniently placed the classifier's own group at the top. None of this was done with genetic evidence, because genetics as a science didn't exist yet. It was done with rulers, calipers, and a great deal of cultural bias dressed up as objectivity. For centuries, this framework didn't just sit quietly in textbooks. It justified slavery. It justified colonization. It justified immigration laws, marriage bans, and forced sterilizations. Entire legal systems were built on the assumption that race was a fixed biological reality, as real and as measurable as height or blood pressure. By the time the 20th century arrived, the idea felt less like a theory and more like common sense, something so obvious it barely needed defending. Which is exactly why, when scientists finally got the tools to test it directly, the results landed with such force. It's worth remembering just how far this so-called science was taken. In the late 19th and early 20th centuries, the idea of race as a fixed biological hierarchy gave rise to the eugenics movement, a body of pseudoscience that claimed entire populations could be ranked by genetic worth. Eugenicists pushed for forced sterilization programs, restrictive immigration quotas, and marriage laws, all justified by the claim that racial traits were passed down as fixed, unchangeable biological packages. These weren't fringe ideas whispered in the margins of academia. They were taught in university lecture halls, cited in courtrooms, and written directly into national policy across multiple countries. It took the horrors of the mid-20th century, and decades of mounting scientific evidence afterward, to slowly dismantle the credibility of that framework. But by then, the assumption that race was a hard biological fact had already sunk deep into medicine, law, and everyday thinking, and it would take the tools of modern genetics to finally test it properly. The first real cracks in the old model appeared decades before anyone had ever sequenced a full human genome. In the early 1970s, a geneticist named Richard Lewontin ran an experiment that, on paper, sounds almost too simple to matter. He gathered genetic data on blood groups and proteins from populations around the world, then measured how much of the variation in those genes could be explained by the racial categories people were using at the time, and how much came from differences between individuals within those same categories. If race really were a deep biological reality, you would expect most of the variation to show up between the racial groups themselves. That is not what he found. Instead, this finding, now often referred to as Lewontin's paradox or Lewontin's fallacy, depending on which scientist you ask, kicked off decades of debate. Some researchers argued the pattern proved race was biologically meaningless. Others pointed out that even small between-group differences can still be enough to statistically sort people into clusters if you have enough GENET, IC markers to work with. That debate never fully went away, but the core finding held up again and again as the data improved. Human beings are, genetically speaking, remarkably uniform as a species. The overwhelming majority of the variation that exists among us exists inside every population on Earth, not carved cleanly between them. Then came the moment that changed everything. In the early 2000s, after more than a decade of work and billions of dollars in funding, the Human Genome Project finished mapping the roughly 3 billion base pairs that make up human DNA. When the results were announced, one number stood out above all the others, and it has been repeated so often since that it's become almost a cliché. Any two human beings, no matter where their ancestors came from, share about 99.9% of their genetic code. That leaves just 0.1% of our DNA to account for every visible difference between us: height, eye color, skin tone, hair texture, disease resistance, and yes, the traits we've historically used to sort people into races. It is an almost unbelievably small sliver of biological real estate to carry the entire weight of what societies have spent centuries treating as a fundamental, unbridgeable divide between groups of people. The Human Genome Project was only the beginning. In the years that followed, larger and more detailed efforts picked up where it left off. The International HapMap project cataloged patterns of genetic variation across multiple populations. And later, the 1000 Genomes project expanded that work dramatically, sequencing the genomes of thousands of individuals from populations across Africa, Europe, East Asia, South Asia, and the Americas. More recently, massive genomic databases like GnomeAD have pooled together genetic data from hundreds of thousands of individuals worldwide, giving researchers an unprecedented, high-resolution view of exactly how human genetic variation is distributed across the globe. Each of these projects, built independently, using different methods, different populations, and different technology, arrived at the same underlying picture. Genetic variation is enormous within every population, modest between neighboring populations, and never organizes itself into the small number of sharply bounded groups that the old racial model predicted. What scientists did next was even more revealing than the 99.9% figure itself. They asked a very specific question. Within that remaining tenth of a percent, does the variation actually cluster into the racial categories we use in everyday language? Categories like black, white, Asian, and so on? Using increasingly powerful sequencing technology, researchers began comparing thousands of genetic markers across populations from every inhabited continent. If race were a true biological category, you would expect to find clear genetic boundaries, sharp lines, where one group's DNA stopped looking like another's and started looking distinct. Instead, what they found again and again was something very different, a gradient, a slope. Geneticists call this pattern Clinal variation, and it means that as you move geographically from one population to a neighboring one, genetic differences increase gradually, not suddenly. There is no wall. There is no fence. There's only a slow, continuous slide across the map of human ancestry, and where you choose to draw a line on that slope is a cultural decision, not a biological one. This becomes even clearer once you understand where all of that variation actually came from in the first place. Every non-African population on Earth, today, descends from a relatively small group of humans who migrated out of Africa somewhere between 50 and 70,000 years ago. That single historical bottleneck had a massive effect on global genetic diversity. Because only a subset of Africa's population left the continent, the genetic diversity found outside of Africa is essentially a smaller slice carved out of the much larger diversity that already existed within Africa itself. In practical terms, this means African populations today carry more genetic diversity on their own than the rest of the entire human species combined. A pair of individuals from two different ethnic groups within a single African country can, in some cases, be more genetically distinct from each other than a person from Europe is from a person in East Asia. That single fact alone dismantles the old idea of race as neatly as anything in modern science, because it means the deepest, oldest divisions in human genetic history exist within a continent that older racial models used to lump together as a single uniform category. Researchers have used this kind of data to build detailed maps of human migration and relatedness, and the picture that emerges looks nothing like the old racial charts from anthropology textbooks. One large-scale genetic study processed more than 65 million rare genetic variants and identified over 17 million shared fragments of DNA passed down between individuals, fragments so old in some cases that they could be traced back roughly 200,000 years. From that data, the researchers identified around 9 broad geographic regions of genetic relatedness, covering areas like East and West Africa, Northern Europe, East Asia, Oceania, South Asia, the Middle East, and South America. Even these 9 regions were not hard, isolated categories. The study found extensive overlap and shared ancestry between neighboring populations, along with heavy admixture in regions like Central Eurasia, where waves of migration, trade, and conquest mixed genetic lineages together over thousands of years. The overall picture wasn't one of separate, self-contained human types. It was one continuous, interwoven web of ancestry, shaped far more by geography, migration, and history than by anything resembling the 4 or 5 races most of us grew up hearing about. So if race isn't really a biological category, why does it feel so real? Why do people from different parts of the world so obviously look different from one another? This is one of the most common objections raised whenever this research gets discussed, and it deserves a real answer, not a dismissal. The visible traits we associate with race, skin tone, hair texture, eye shape are real, and they are genetic. Nobody is claiming otherwise. What the research shows is something more specific. those particular traits are controlled by a very small number of genes, and those genes happen to be under unusually strong evolutionary pressure from environmental factors like sunlight exposure. Skin pigmentation, for example, evolved repeatedly and independently in different human populations as a response to how much ultraviolet radiation an environment received. Populations that lived for many generations near the equator tended to evolve darker skin, which protects against UV damage and folate depletion. Populations that migrated to regions with weaker sunlight tended to evolve lighter skin over time, which allows the body to produce enough vitamin D. This is a real, well-documented evolutionary process, but it is also a highly localized one. The genes responsible for skin color do not reliably track with the genes responsible for, say, disease resistance or bone density or blood chemistry. In other words, skin color is a poor proxy for the rest of a person's genome because it responds to a very specific environmental pressure that has nothing to do with most of the rest of human biology. This is actually the central insight that geneticists keep returning to. A handful of visible traits, the ones humans have historically used to sort each other into racial categories, are controlled by relatively few genes that evolved quickly and visibly because of environmental pressure. But the vast majority of the human genome, the genes tied to internal biology, disease risk, metabolism, and countless other traits, don't sort along those same lines at all. Two people with similar skin tones from different continents can carry wildly different genetic profiles for traits that have nothing to do with pigmentation. Meanwhile, two people with very different skin tones can share close genetic similarity in traits that actually matter for medicine. The visible markers we've built entire social systems around turn out to be genetically shallow, a thin surface layer sitting on top of a much deeper and far more complicated reality underneath. This disconnect has enormous consequences once you move from abstract science into the real world, particularly in medicine. For decades, doctors were trained to use race as a rough proxy for genetic risk, assuming that a patient's racial background could reliably predict things like drug metabolism, disease susceptibility, or kidney function. Modern genetic research has increasingly shown this approach to be deeply flawed. Diseases like sickle cell anemia, for example, are often associated with black populations in the public imagination, but the trait that causes it, a mutation that offers resistance to malaria, is found in populations from parts of Africa, the Mediterranean, the Middle East, and South Asia alike, because it evolved independently wherever malaria was historically common. It has nothing to do with race as a category and everything to do with a shared environmental pressure across very different populations. Relying on racial assumptions in medicine has, in numerous documented cases, led to misdiagnoses, delayed treatment, and unequal care, precisely because race is such an unreliable stand-in for the genetic and environmental factors that actually determine health outcomes. The same misunderstanding shows up constantly in popular culture, particularly around sports and athletics, where people often assume that certain populations are simply built for certain physical feats because of race. The genetic reality is far more specific and far less tidy. Traits like fast twitch muscle fiber composition, lung capacity, or bone density are influenced by particular genes that can be more common in some regional populations than others, largely due to localized evolutionary pressures, and, just as importantly, training, culture, and opportunity. But those genes do not track along racial lines the way popular assumptions suggest. Elite distance runners from certain highland regions of East Africa, for example, share specific genetic and physiological adaptations tied to their particular altitude and environment, adaptations that are not shared by other populations, casually lumped into the same broad racial category. Treating an entire race as athletically uniform collapses an enormous amount of real, fine-grained genetic and environmental variation into a crude stereotype that the actual data simply does not support. Forensic science has run into a similar wall. Investigators have sometimes used DNA phenotyping, predicting physical traits like skin tone, eye color, or ancestry from a genetic sample, to help generate leads in criminal cases. This technology can be genuinely useful, but it works by estimating probable ancestry and visible traits from specific, well-understood markers, not by identifying someone's race in the traditional sense. The industry itself has increasingly moved toward describing results in terms of ancestral population percentages rather than racial labels, precisely because the older racial categories are too imprecise and too culturally loaded to serve as reliable scientific descriptors. Even in a field built around extracting maximum information from a tiny biological sample, race turns out to be the wrong tool for the job. None of this means ancestry is meaningless, and this is a distinction that tends to get lost whenever this topic comes up. Ancestry, in the genetic sense, is real and measurable. If your recent ancestors came from a specific region, you will likely share detectable genetic similarities with other people whose ancestors came from that same region. And that information can be useful, particularly in medicine, historical research, and even criminal investigations. This is exactly why consumer genetic testing companies can tell you that your ancestry traces back to particular regions of the world with a reasonable degree of confidence. But ancestry is not the same thing as race. Ancestry is granular, specific, and continuous, tracing back to particular regions, migrations, and family histories. Race, as it has traditionally been used, is a blunt social categorization system that lumps huge, genetically diverse populations together based primarily on a small number of visible traits. You can have detailed, medically useful ancestry information about a person without that information ever needing to pass through the crude filter of racial categories at all. Modern population geneticists have largely moved away from using the word race in their research entirely, replacing it with more precise language like ancestry, population, or ethnic group, specifically because these terms map much more accurately onto the genetic patterns that actually exist. Major scientific organizations, including large professional bodies representing human geneticists, this isn't a fringe position or a recent trend driven by politics. It reflects a slow, decades-long convergence of evidence from genetics, anthropology, and evolutionary biology, all pointing in the same direction, arrived at independently by researchers working in different countries, different institutions, and different decades. Consider, too, just how recent the tools behind all of this discovery actually are. Researchers only gained the ability to sequence a complete human genome quickly and affordably within the last 2 decades. Before that, scientists were essentially arguing about the biological reality of race using indirect evidence, blood types, skull measurements, and small samples of protein markers. It's only now, with genome sequencing available at a scale that lets researchers compare tens of thousands of individuals across every populated continent, that the full picture has finally come into focus. Which means this isn't an old debate that's finally been settled. It's a very new, very detailed answer to a very old question, and the data keeps arriving faster than most people have had time to catch up with it. It's worth pausing here to be clear about what this research does not claim, because misunderstanding this point has fueled a lot of confusion and bad faith argument online. This isn't a claim that human populations are all identical or that geography and ancestry have no biological consequences whatsoever. Populations that have lived in specific environments for long stretches of time have absolutely adapted to those environments in measurable genetic ways. From lung capacity at high altitudes in the Andes and the Himalayas, to lactose tolerance in populations with long histories of dairy farming, to malaria resistance in regions where the disease has been endemic for thousands of years. These are real, well-documented, published findings. What the research pushes back against is something narrower and more specific: the idea that humanity naturally divides into a small number of discrete, internally uniform, biologically bounded races, each with its own package of traits that neatly separates it from the others. That specific model, the one that shaped centuries of law, science, and social hierarchy, is the one that hasn't held up under genetic scrutiny. The messier, more accurate picture is one of continuous variation, deep shared ancestry, and traits that don't travel together the way the old racial categories assumed they did. There's another angle to this story that rarely gets discussed outside academic circles, and it involves how scientists actually try to statistically cluster human populations using genetic data. In the early 2000s, researchers ran a series of studies using computer algorithms designed to sort individuals into genetic clusters based purely on patterns in their DNA, without being told anything about their self-identified race or ethnicity beforehand. Depending on how many clusters the algorithm was told to look for, the results could roughly correspond to broad continental groupings. But this outcome was heavily dependent on the specific population sampled and the number of clusters requested by the researchers themselves. Ask the same algorithm to sort people into 3 clusters, and you get one picture. Ask it for 5 or 10 or 20, and you get an entirely different, more finely grained picture. This became one of the most important methodological lessons to come out of the entire debate, that you can make human genetic variation look like discrete races if you go looking for a small number of categories, but that same data can just as easily reveal a smooth, continuous gradient if you don't force it into a predetermined number of boxes. Twin and family studies add yet another layer of nuance that's worth understanding here. Researchers have spent decades studying identical twins who share virtually all of their DNA to figure out how much of a given trait is influenced by genetics versus environment. This research has consistently shown that traits like height, certain disease risks, and even aspects of temperament have a meaningful genetic component. But critically, this heritability research operates entirely at the level of individuals and specific traits, not at the level of races. Knowing that height is partly heritable tells you nothing about whether one racial group is taller than another for genetic reasons, because average differences between populations can be driven overwhelmingly by environmental factors like nutrition, healthcare access, and historical circumstance, even when the trait itself is highly heritable within any given family. Conflating individual heritability with between-group difference is one of the most common and most persistent errors in how the public understands genetics, and it's a distinction that geneticists have spent enormous effort trying to communicate clearly. There's also a deeper, almost humbling layer to all of this once you zoom out to the scale of the entire human story. Every single person alive today, no matter their appearance, their language, or their country of birth, descends from the same relatively small population of early humans who lived in Africa roughly 200,000 years ago. In evolutionary terms, that is an almost impossibly short span of time. The visible differences we've spent so much energy sorting each other by, skin tone, hair texture, facial structure, are, on a genomic scale, incredibly recent and incredibly shallow modifications layered on top of a shared, common foundation. We are, quite literally, one of the most genetically similar large mammal species on the planet, a fact that surprises most people the first time they hear it. Populations of chimpanzees living in neighboring forests in Africa often show more genetic diversity between them than exists across the entire global human population combined. Our species went through such a dramatic population bottleneck at some point in our evolutionary history that we never had the time, isolation, or population size needed to develop the kind of deep genetic divergence that would be required for biologically distinct races to actually form. So where does that leave us, standing here with all of this evidence laid out? It leaves us with a picture of humanity that is, in many ways, far more interesting than the old one. Instead of a small number of fixed, separate categories, we are one continuous, deeply interconnected species, shaped by tens of thousands of years of migration, adaptation, mixing, and shared history. Every population on Earth carries fragments of that shared story written directly into its DNA, fragments that can be traced, mapped, and studied with a level of precision that earlier generations of scientists could never have imagined. The old framework of race told us we were fundamentally divided. The genetic evidence tells us something closer to the opposite, that beneath the surface differences we've spent so long focusing on, we are overwhelmingly variations on the exact same underlying blueprint. What the genetic research does is separate two things that were, for centuries, treated as one and the same. It shows that the social and historical weight carried by racial categories was never actually backed by the kind of deep biological division people assumed was there. The boxes were real in their consequences. They were never real in the way biology actually works. It's also worth acknowledging how strange it is that this conversation still feels controversial to some people, given how solid the underlying evidence actually is. Part of the reason is that the old racial categories are still deeply embedded in everyday language, in census forms, in medical intake questionnaires, in casual conversation. We are, understandably, slow to let go of categories that feel so intuitive, so visually obvious, so baked into how societies have organized themselves for generations. But intuition has been wrong about biology before, and often spectacularly so. For centuries, it felt intuitively obvious that the sun revolved around the Earth, that heavier objects fell faster than lighter ones, that diseases were caused by bad air rather than invisible microorganisms. In each case, careful, patient scientific investigation eventually overturned an idea that had felt like plain common sense for generations. The genetic story of race is following a similar arc, a slow, sometimes uncomfortable correction of an idea that once seemed too obvious to question, replaced by a more accurate, more complicated, and ultimately more truthful picture of what we actually are. And that, in the end, is the quiet, world-altering discovery sitting inside all of that genetic data. Not a dramatic twist, not a single shocking headline, but a slow, patient accumulation of evidence, built study by study, genome by genome, over more than half a century, all pointing toward the same conclusion. The lines we drew between races were never written into our DNA. They were written into our history, our institutions, and our assumptions. Our genes, it turns out, were telling a very different story all along, one of a single, deeply connected human family spread out across every continent on Earth, carrying the same ancient code, shaped by the same long migration out of Africa, bound by the same shared history with only the faintest, thinnest layer of variation, separating any one of us from any other, no matter where on the map our particular story happened to begin.